Good afternoon. It's now my pleasure to introduce Kevin Tan, CFO, Paul Herzig, CTO, and Brian Collins, SVP at Solid. Thank you all for joining us today. The format for this session is roughly a 25-minute fireside chat. And if anyone has any questions, you can ask them in the question box. And to start off, perhaps you could give us an overview of Solid's CMC capabilities and how they fit into the company's overall strategy and the overall approach going forward.
Absolutely. Thank you so much for having us. And we're so pleased to be participating in the Chardan conference and the manufacturing summit. I think I'll start off first by, just reiterating that there may be forward-looking statements made and we may need to disclaim any obligation to update them in the future, even if they do change. I think it's important to orient everyone around Solid as a new company. In December 2nd of 2022, we completed a merger with Avanti Biosciences and combined both companies' pipelines. People here might remember Solid as being a Duchenne-only company. That's not the case anymore. We have a Duchenne asset that's our most advanced asset that should be entering the clinic in the second half of this year. Having said that, we do have an asset in FA as well as in a growing cardiac pipeline with three candidates that we're currently working on. We have a very deep and experienced management team. When you look at it, between Bo, Cumbo, Ty, and myself, all with experience at Sarepta, not just in genetic medicine, but in rare disease specifically. I want to kind of point out, Paul, who is on this call, as being one of the leading experts in gene therapy CMC with experience, not just at Solid today, but previously at Avanti Bio, where he came with Bo and Ty, but also Bridge Bio, Logic Bio, Novartis. Jesse, who's a regulatory expert with really deep relationships with the FDA. We have CMC regulatory experts on staff. So really from top to bottom, it's a very deep management bench. As we look at this, we believe that we're a true platform genetic medicine company. We have our own vector core down in North Carolina. We're able to manufacture our own constructs. We have a growing capsid library, and we really believe we're kind of fit for purpose in terms of kind of being a platform genetic medicine company. And this really kind of calls out why we're here, which is our expertise in gene therapy manufacturing. Beyond as I called out the vector core, we have within 100 feet of where I'm sitting, 50,000 square feet of lab space where we can scale from 2 liters up to 500 liters. We took our experiences at Sarepta and really put CMC at the core of this company so that we can control the PD, manage the scale up, speed constructs through preclinical work, and own the IP. Importantly, one thing that we don't often kind of point out, but we do have, is something called MS&T, which is manufacturing science and technology. We have a team right now sitting with our CDMO manufacturing partners, and they're embedded with the team there to ensure that we are able to tech transfer the asset over to the CDMO. We aren't sending the steps and crossing our fingers. We're making sure that the process is transferred appropriately, and we really believe that although it's higher in headcount, it really pays dividends in terms of improving our yields and preventing failed batches. And then this really kind of speaks to, we did change our manufacturing process for our current candidate from HSV to transient transfection, and we did see significant improvements in microdystrophin expression from that manufacturing change. And then this kind of calls out our growing pipeline, not just in neuromuscular, but also in cardiac. And as you can see, we're not a single asset company anymore. We have many shots on goal that are developing. And then lastly, this kind of calls out the milestones that we anticipate this year with an IND in the second half of this year and into the clinic as well in the second half of this year. We do have $213.7 million in cash as of end of the year last year. which we're guiding the street to take us into 2025.
Great. Is that all, or do you have one more slide?
That's it.
Perfect. Yeah, so you mentioned that Solid has transitioned programs from HSV to the transient transfection processes. Can you elaborate a little bit more on that and walk us through the factors underlying that decision?
Yeah, I'll let Paul maybe speak a little bit more about the manufacturing change and kind of the rationale behind it.
I think I'll actually jump in and take that one. So thanks, it's a great question. And obviously a number of factors went into the consideration to make a strategic pivot away from HSV to TT. Amongst those include the considerations around COGS and speed. Producing a DNA plasmid is faster, cheaper, and has considerably more available suppliers than to use an HSV as a starting material. Second thing is robustness. Technology for plasmid production has been around for quite a while. And as such, the industry has been able to develop approaches to assure a consistent, pure supply of that plasmid starting material. And then finally, as Kevin alluded to in one of our slides, with the TT material, we've seen a greater level of transduction efficiency in our DMD asset as we transition from HSV to TT. So roll those three things together, you've got a fairly strong argument to do the pivot from HSV to TT.
Mm-hmm. And can you talk about what you've learned from working with these different processes and also different capsids, as well as from prior experience in the clinical gene therapy space?
Yeah, I'll take that one again. To Kevin's intro, you look at Paul's experience as well as a number of other members of the team. We do have a deep history with working with precision gene therapy medicines, in particular with AAV. But each platform, be it TT or HSV or BEV, has its own unique challenges that go along with it, of course. But key to all these things is probably really the core understanding that the starting material needs to be established correctly from the get-go. So as an example to that, really locking in and understanding your promoter sequence, the appropriate structure of your gene construct is critical to get right that first time. There's no going really back and redesigning that. You need to have that ready at the beginning. On the capsid front, from a manufacturing perspective, it's actually not the challenges around productivity that's really been a key learning for us. That's really built into the way we design the capsid and the way that we screen for that construct. So by the time you get to manufacturing, we've really built in a robust understanding such that you have an assurance of a significant manufacturing yield. But the real consideration is actually, as you transition to these novel capsids, is making sure that you've built in the appropriate analytical strategies. Because now you're looking at unique attributes that you might not have as compared to a traditional AAV9 or an AAV2. So really building upfront your analytical approach that will parallel then with your manufacturing development as well. That's been just a couple of the considerations that we've really taken away from working across this breadth of manufacturing strategies.
Mm-hmm. And so in terms of scale, what is your current process scale, and to what extent do you expect that to be sufficient for commercial use across your different programs?
That's a great question. I'll take that as well. Currently, we work at 1,000 liter as our manufacturing scale, which should be sufficient for us from a commercial standpoint. Our CDMO partner does have the capability and definitely the interest of going all the way up to 5,000 liters, should we require that.
Great. And so you mentioned analytics earlier. Can you elaborate a little bit in discussing your approach to assessing the purity, potency, and the other quality attributes of your gene therapies?
Absolutely. Again, I think it's kind of two arms to that conversation. The first is really leveraging the historical experience in understanding the relationship between product quality and safety and efficacy. And that's really a key foundation to building up a robust analytical approach. Then you parallel on top of that a real investment in Solid into novel technologies to support the analytics and the analytical development. If you look at the capsid itself, there's a lot that goes into just the decorations that occur on the outside of the viral particle that can be impactful with regards to the efficiency of transduction or infection. So one of the things we definitely spend a bit of time on is developing cutting edge technologies to be able to look at all those types of modifications on the cell surface, leveraging mass spec and otherwise. As an industry, we've always been interested in also understanding how many of those capsids contain our gene of interest. Everyone's talked about the percentage of full versus the percentage of empty. Still a critical component that one needs to keep an eye on during the course of developing in the manufacturing process. But now it's really moving past that. It's not only asking the question, is the gene present, but is that gene that's present full length? Is it broken at all? Are there any other fragments of DNA from the plasmid or the host cell line that might be packaged along with that? And to support that, it's really doing an investment in gene sequencing technologies, next-gen sequencing or the otherwise, that has really allowed us to gain a better insight into specifically what you've got in your gene therapy and assure that you've got a very safe and efficacious product.
Got it. And so from your perspective, how clear are the FDA and the other regulators' expectations regarding what they're looking for with respect to CMC?
Sure, I'll take that one. Excuse me. What I would say is the expectations are becoming clearer each and every day, right? What we've seen is it's not different than many other drug development pathways, except AAV has been, that specific approval pathway is being defined as we advance, right? The approval of Luxturna and Zolgensma have kind of laid out a pathway, and everyone is continuing to build on that and advance the industry forward.
Got it. And you talked a little bit about the next-gen capsids that you're working with. Can you expand a little bit on how CMC and manufacturability tie into the capsid discovery work that Solid is doing?
Yeah, so what we do is we screen productivity and packaging very early on. We've established a vector core in the RTP area where we can produce a large number of constructs in parallel and really screen and evaluate that. It's a scaled down version of our clinical manufacturing process, using the same cells, the same downstream and upstream unit operations. And it really allows us to kind of predict how that capsid with a known GOI will compare to other constructs that we've created in the past. And we can just do a high level statistical analysis and say, yes, based upon this, we know we can get 20%, 30% out more when we scale it up. So it allows us to really kind of set that as a criteria before we even take it into preclinical type work.
That makes sense. And just in terms of the process itself that you're using, is that a suspension process?
Yeah, so we have standardized on a suspension HEC 293 triple transfection process is what we've been moving forward with.
Got it. And so I think in the current financing climate, there's a lot of discussion about costs and issues there. How is the current financing climate influencing Solid's decisions regarding what to invest in on the CMC front?
Yeah, so we feel that the CMC needs to be a primary focus by any gene therapy company. If you look across the industry, a lot of the delays and the regulatory issues have traditionally been caused by CMC issues. Making the correct investments early on and owning our science, as well as our manufacturing, technologies will hopefully pay dividends longer term and, eliminate any potential clinical holds, and hopefully allow us to advance rapidly towards licensure.
And I can actually add one thing to that. From our experiences, definitely what we've found is that, and as I alluded to, CMC has been put at the core of this company. Between having our own vector core to speed the development of preclinical materials, not only does it save time, but it saves us money. Beyond that, by having the ability to own our scale up internally, we really found, that, we're positioned to drive COGS down as we manage the yields up. So, by taking that out of the hands of the CDMO and controlling it on our own, we really believe we're positioned well to, have cost-effective gene therapies in the future, but we've made the right investments today.
Got it. That makes sense. So what do you see as the major challenges with respect to CMC in the gene therapy space?
I'll go ahead and take this one. I think the exciting field of CMC and gene therapy is what really brings a lot of us to it. It's bringing in aspects of protein, biologic therapies, other genetic therapies, as well as even higher molecular analyses, all together in a single molecule, which makes both an exciting and complex puzzle to build from a manufacturing standpoint and characterize and tease apart from an analytical standpoint. But on top of that, I guess, the other challenges are what most of us in the industry face around setting realistic expectations. With gene therapy here, we have the challenges around establishing proper starting materials, developing a process, a robust process, a manufacturable process. And how do we do this in a manner that's fast enough to support the expanding needs of our patient population, the new indications that come along that really need these cutting-edge gene therapies? So compressing all that development down to as fast as we can to develop and deliver a safe and efficacious product is one of the key challenges for our industry.
Got it. And so on the flip side, are there innovations that you're excited about that can either address the challenges or kind of take the manufacturing in a new direction?
You ask anyone on my team, I get excited about a lot of things in science, but I guess, there are two areas. One, from a manufacturing standpoint, there's been a recent investment in really understanding some of the fundamentals in how do you assemble a viral particle, and taking that now and translating this into novel molecular tools for the efficient manufacturing of this product is really what's exciting. Now you're seeing the fruits of the labor that are transitioning from the research field into the development and manufacturing space. And then the second thing is around really the analytics I referred to. The tools that we have now to dive deep and look at much more of a molecular level. What does that viral particle look like? How do we assure the quality to be consistent and pure? Those two things together, I think we're at a great point in the gene therapy field where we're going to see some very substantial breakthroughs in the next couple of years.
So on those topics, I guess, as we get greater understanding of what it takes to build a viral particle, and as the analytics tools continue to develop, to what extent do you expect standards for what a gene therapy should be to kind of evolve, as from either a regulatory standpoint or from a functional standpoint?
I guess I could take a lead on that one. Mm-hmm. I imagine that we're going to see a lot of the evolution we've observed previously for the protein therapeutics. If you look at the early days of protein therapeutics, it's a very different space than where we're at now, with an expectation with regards to the depth of the characterization and standardization, if you will, of some platform technologies. I expect that we'll see the same type of evolution from a gene therapy standpoint. So when we look 10 years from now, things that we may be considering as bespoke or as incredibly novel may become a commodity in the next 10 years, which will really be just an amazing advancement for our field, and quite a gift to the patient population as well.
Yeah, we've had a number of conversations how the AAV space is very similar to the late 90s from a monoclonal space at the end of the day. And now, it's a fairly robust transferable process once people have done the science and understand what it takes to actually get there at the end of the day.
Got it. So what do you think people might be missing regarding Solid's current manufacturing capabilities as the company is in 2023 after its evolution that you've gone through?
I'll throw a couple things in there. What we've really experienced is, bringing these two companies together. What we didn't realize perhaps prior to entering into this is the way the two pieces can come together and really add great value beyond just the sum of the two parts. There's been an amazing synergy between the two teams, a similar line of thought with regards to experience, but the depth of understanding is very different in the two organizations, which really allowed the two to come together to be a much more efficient manufacturing and development machine, if you will.
Yeah, I think that beyond kind of the nuts and bolts in the CMC team, I think that what people might be overlooking is the focus on CMC, not just from a kind of analytical process development and quality perspective, but also having the right CapEx and the right points in the process. And then also having a focus on CMC reg, which has been, kind of a speed bump for development of assets for other companies, and really being kind of focused on CMC as being kind of a true kind of competitive advantage for Solid Biosciences versus our peers.
And one other thing I'd like to add, right, is the acquisition, of Avanti brought, two distinct organizations together, which had individual strengths, right? Solid had a really strong analytical background and a extensive knowledge on HSV, AAV manufacturing, whereas Avanti came into the organization with a really strong background in triple transfection. And as we pivoted the platform, that kind of helped lift the entire CMC organization up and continue to progress things forward faster.
I think the last thing I'll say is a fearlessness around asking more questions, diving deeper in better understanding both our process and our product. That's something that maybe outside of the organization you don't have the transparency into.
And one last question in the last couple of minutes. In recent years, the field has seen considerable supply chain constraints that have affected manufacturing timelines in the cell and gene therapy space. Just wondering what you're seeing these days and how a Solid approaches supply chain for its programs.
Yeah, so in general, right, the supply chain constraints over the last couple of years have been predominantly driven by COVID. From two different fronts. First, that sucked up a lot of the general viral manufacturing capacity out there. And second, throughout the entire supply chain, critical starting materials, raw materials, and consumables were scarce if at best. What I would say is at this point, you're seeing lead times for critical raw materials come way down. A lot of the critical suppliers have increased their manufacturing capabilities and built inventory. The demand for COVID vaccines has kind of equilibrated across the industry, and everybody seems to kind of be, lead times that had gone to a full year back down into the 12 to 16 weeks, which is where they were at pre-COVID at the end of the day. And then from an AAV manufacturing capacity, CDMOs have continued to build out capacity, and the 12 to 16 month lead times have started to come back down to be able to kick off programs.
Great. So I see we're just about at the end of the session, so I'd like to thank all of you again for your participation.
Thank you
so much
for having us.
Thank you.